Hello, my name is Amy Cheung, and I'm a Senior Director of Integrated Drug Development at Certara. I'll be presenting Module 4, Case Study 4, Vaccine, MIDD, and Beyond. The objective of this module is to outline the challenges of vaccine development, especially with the rapid progression of the COVID-19 pandemic. We also want to highlight the usefulness of MIDD in vaccine development by demonstrating the importance of knowledge building through model interconnectedness of a compound to support vaccine drug development. A few examples selected from the literature to demonstrate the application of modeling approaches to understanding immunogenicity and its relation to efficacy, and how modeling framework can guide how infection transmission translates to economic impact. A vaccine is a type of therapeutic that perturbs the body immune system in a strictly controlled way, which is subsequently produce memory protections so that it can fight a disease it has not been encountered before. Vaccines are designed to prevent disease as prophylactic, rather than treat disease once you have caught it. Many of you have seen this graphic illustration on the left. Beyond a doubt, vaccines are the most effective medical intervention for saving human lives and preventing suffering. However, that doesn't mean we always get it right. Much focus is logically aimed at assuring vaccines are safe in large populations. We need to make sure that essentially healthy people exposed to a vaccine do not experience unwanted safety risks. Unfortunately, the track record on efficacy is less strong. For instance, seasonal influenza, which is shown on the right and the middle diagram. Each year, we race to create a new vaccine to address the latest permutation of influenza strain, and these vaccines are typically safe and well tolerated. However, efficacy, effectiveness in preventing the flu, hospitalization, and death differ from year to year, partially because seasonal differ and new strains may be more virulent, but also vaccines may be less successful in triggering the lasting immune response to protect us effectively, which in turn undermines herd immunity levels. In the painting here, you can see Dr. Edward Jenner, who administered the first vaccination as a preventive treatment for smallpox, a deadly disease that had killed many people over the centuries. Since then, Jenner's model was employed by many scientists through the 19th and 20th centuries to develop new vaccines, such as whooping cough, hepatitis B, and many more shown in the blue box here. The method for developing and understanding the vaccine mechanism of action has improved over time. Commonly, a vaccine is dosed on animals to ensure safety prior to human trials. In a non-clinical experiment, a starting dose is tested and escalated until the dose is no longer safe or saturated. This is then provided a safe dose range to be scaled up to applied in clinical settings. In humans, the dose decisions are made based on escalation results and pairwise comparison of the best dose. In addition, if a next-generation vaccine is available, a higher sample size might be required to allow the comparison of the vaccine. The time required for the stepwise approach from vaccine candidate to approval is long. Much information and data are then left behind once the vaccine development has moved forward or terminated. Development programs logically focus on safety foremost. If we are going to expose billions, we better make sure it is safe always. We also note that to accelerate development of dosing strengths and regimens are not tested widely. By the time one gear up for phase 3, one needs to know what the large-scale manufacturing should look like. The dose and regimen get locked in early on, thus after phase 1 with safety and tolerability, then followed by phase 2 with safety, tolerability, and immunological response. We typically then enter phase 3 with prophylactic efficacy with a single dose strain. However, there are many questions remain. For example, do we have the right dose and right regimen? Is it optimized for a lasting immune response? Do we need to revaccinate, and how often? Do different populations have different dosing needs? They're all critical questions. Just like there is an ongoing race to repurpose existing drugs to actively treat COVID-19, we now to see the dose selection being lower priority. There's a risky strategy, but accelerating the vaccine for COVID-19 should not be about creating a safe vaccine very quickly, but rather a safe and efficacious vaccine as quick as possible. And getting it right the first time instead of the second, third, or fourth shot is the goal, because the production cost and the medical urgency simply don't allow for multiple shots on a gold approach. And all of this consideration will also allow us to prepare future pandemic situations. The key questions for vaccine development center on immunity and protection, dose and regimen, competitor landscape, and target populations. For example, what level of immunogenicity is required to achieve protection, and what is the corresponding dose and regimen? How many booster vaccines are required and how often to ensure lasting protection? Using COVID-19 as an example, which targets the whole global population for 0 year plus, therefore, the understanding of whether maturation or deterioration due to age impact immunogenicity and protection is essential, in particular for pediatric, elderly, and vulnerable populations. In addition to immunogenicity, markers such as IgG titer or concentration. What are the biomarkers, for example T-cell, immunomarker, C-reactive protein, interferon gamma, innate immune system, C5 peptides, are related to response or efficacy or the outcome? And can we utilize information from emerging trials and from the literature? Vaccines have been developed to protect people from viral, bacterial, and parasitic infections. From a model-based point of view, the analysis approach are very similar. We can make use of the wide range of data reported in the literature to understand the adaptive immune system. We will illustrate the model-based approach just described using several examples of different antiviral vaccines and demonstrate how it can be applied to, for example, COVID-19 or other virus listed here. However, before I do that, I will refresh and introduce some MIDD concepts and approaches. As mentioned, MIDD approaches can enhance vaccine development. With this framework, key questions are set at the beginning, and these questions are continuously answered as information emerges. 3 levels of question can be asked to understand the disease, the vaccine, and the mode of action. And sometimes due to the novelty of many viruses and vaccines, assumptions and data gaps must be identified to quantify the uncertainty. This assumption must be tested and validated as information emerges. MIDD also offers a range of modeling approaches such as model-based meta-analysis, MBMA, and quantitative system pharmacology, QSP, to accelerate vaccine development. One of the most important questions on vaccine development is the level of immunity required to establish protections and the duration of protection. This is a function of dose and when booster vaccine doses are administered. To answer this question, the understanding of immunological memory is essential. On the left, a high-level diagram illustrates how the body's adaptive immune system can learn to recognize new invading pathogens, for example as SARS-CoV-2. This enables us to develop a mechanistic mathematical models on the right to describe how vaccine primes the adaptive immune system, including the generation of antigen-specific memory B cells and the duration of immunological memories. Now I want to highlight why MBMA is useful in vaccine development by giving a general introduction of meta-analysis. The simplest meta-analysis is to generate a pairwise comparison between placebo and vaccines across multiple data sources. This does not enable us to compare different vaccines. To do this, we have to use a network-based meta-analysis where we can do an indirect comparison of vaccines by the differences with placebos. However, this still treats different dose levels of the same vaccines as separate treatments. And so, to infer the dose-response relationship, we have to use a model-based meta-analysis, not only allowing us to take dose level into account, but also covariates such as age, race, and comorbidity or formulations. Together with comparator database and MBMA analysis, we can inform decision-making in vaccine development. This ranges from dose and population selection, go-no-go criteria, to market position using virtual head-to-head comparison. In brief, MBMA incorporates parametric models based on literature data and in-house data. It quantifies the effect of the treatment, time, and patient population characteristics on the outcome. It can include trial-level covariate relationship on the dose-response model to account for trial differences in patient populations. It also allows for simultaneous modeling for multiple endpoints and can therefore link biomarkers, for example immunogenicity, to clinical endpoints, for example incident rate or hospitalizations. Like network meta-analysis, it can provide indirect comparisons and simulations of head-to-head trials, but uses longitudinal dose-response model for individual vaccine or vaccine classes. It can also be used for simulation for trials and trial success predictions. Here we present our MIDD interconnectedness framework that can be applied to vaccine development. In Step 1 and Step 2, a wide range of doses and formulation are tested in non-clinical species, for example in rodent and non-human primate models, to understand immunogenicity over time. Through mathematical modeling, the developed models allow predictions and understanding of the dose-response relationship. Historical vaccines in similar classes can also provide information on dose range. The prediction can also provide recommendations of how best to carry out the next non-clinical experiments to collect missing information to refine predictions and establish confidence. In Step 3, the developed animal model is translated to humans to predict the human dose-response relationship using the human immune response model. In Step 4, doses selected using the model are tested for the first time in man trial. As the clinical data emerges, this is fed back to refine the model. This learning and confirming through modeling informed approach continue as the vaccine moves along the development phases. The established model can also predict the dose and regimen required to achieve protection. In Step 5, public, non-clinical and clinical information of other vaccines in different populations, including animals, pediatric, elderly, and other vulnerable populations, are used to build a database. MBMA uses this data with the data for the investigational vaccines to predict the relationship of immune response and outcome, to inform dose selection and various populations, and allow early head-to-head comparison. And in Step 6, this allows acceleration for go, no-go, and subsequently approval. As mentioned, we will now go through 5 examples, each of them addressing an important question for vaccine development. This example demonstrates the impact that MIDD can have at different phases in the drug development trajectory, such as using non-clinical data, designing an optimal dose regimen, predicting the efficacy of a new vaccine, and extrapolating to long-term protection against viral infections. Please note example 1 and 3 were also presented by Dr. Jeff Sachs at the PAGE Pharmacometrics meeting in 2019. The first example is about making better use of non-clinical data to support translation to humans. Non-human primate longitudinal antibody titer data at various doses was used to develop a model mimicking the adaptive immune system. The model described the dose and time dependency of the antibody titer. In Figure 1, the blue circle are the observations, the black line is the model fit, and the red line represents the geometric mean of the data. You can see the effect of a booster dose causing antibody titer to rise to much higher level, which are well described by the model. The response is saturated between the two dose levels shown. The model was translated to human to allow for predictions of the immune response at different dose levels, regimens, and formulations. This human model was refined with phase 1 data and then used to influence the phase 2 trial design by incorporation of lower dose level and an additional regimen. So the impacts of work include increased confidence in the ability to translate findings from non-clinical species and the ability to model the immune response of virus K vaccine. It also changed the planned phase 2 design. The second example shows how MIDD supports the selection of dosing regimen needed for a required immunogenicity response for hepatitis B virus vaccine. In this example, data from 21 trials with 1,923 adult patients across 8 hepatitis B vaccines were analyzed reflecting over 10,000 post-vaccination antibody titer sample. A fairly simple 3-compartment model was developed to describe the relationship between vaccine administration, memory cell growth, and antibody excretion. The model has 6 unknown parameters, of which 5 had to be estimated. A key assumption made was that the immune memory is assumed to reach a maximum after priming. 1 or 2 booster vaccination administrations may be needed to obtain the full potential of immune memory in terms of anti-hepatitis B antibodies. Simulations of scenarios allowed varying the dose regimen. The plot showed a simulation of a typical study applying 3 20 microgram vaccine dose at month 0, 1, and 6. The precise rate of antigen decay may be relatively unimportant in qualifying the rate of memory cells generation, as long as most of the circulating vaccine antigen is removed prior to the subsequent vaccine dose. The generation of memory cells is found to plateau after approximately 6 months. The left-hand side panel shows estimates of 2 of the parameters related to the rate of memory cells generations for various vaccines included in the analysis dataset, as well as the predicted time needed to obtain protective immunity. The average time to develop memory levels about 10 mil/mil is the best indicator for time taken to develop protective immunity after vaccination. The plot at the right shows model fit of the data. Each curve represents a different study. The 21 different studies with 8 different vaccines and different dose regimens were well described by this relatively simple model. Based on the modeling, it was concluded that a single vaccine dose could be sufficient to generate protective immunity. This could mean that additional immunogenicity trials can be omitted for the development and analysis of new vaccines. If we look at the different levels to design the strategy of vaccination, we need to look at the hepatitis B virus transmissions. Using epidemiology model, we can understand the impact of vaccination transmission dynamics to evaluate different immunization strategies. The epidemiological compartments and definitions are as below. Susceptible is denoted by S, at risk of the infections with HPV. Latent L, which is individuals who have been infected but not yet infectious. Acute A are individuals who are in the initial highly infectious stage of HPV infections. Carrier C, people with chronic HPV infection who are infectious or non-infectious to other. Immune I are individuals who recover from the carrier stage or acute stage of HPV infection or have been successfully immuned recovery are individuals who have recovered from the carrier stage or kill stage of HPV infections. Vaccinated V, individuals who have been successfully immuned. The antibody efficacy PKPD model, which also allows calculation of efficacy, can also provide the probability of projections or immune to this model. This also highlights the importance of incorporation of the force of infection parameters, which is defined as the probability per unit of time that a susceptible individual become an infected individual in a simulation. In the table here shows how different types of models are based on the availability of data and also, for example, based on the n number of trials, and the evaluations of the prediction are also shown in this table. In our third example, we predict the efficacy of a new vaccine X. First, literature data from clinical sources were collected. This data, together with the in-house data, were used as input to develop an MBMA model, which included both vaccine and placebo data. Optionally, we could have included non-clinical data to support model development. The MBMA model described the relationship between immunogenicity biomarker and clinical outcome based on immunological understanding. In this case, the clinical outcome was the incidence rate of the occurrence of a viral infection. Covariates were included in a model to describe differences among populations, route of administration, the severity of disease, and formulation options. In the next step, the resulting model was used to simulate a large population with and without the vaccine. Then the efficacy was calculated based on the resulting predicted incidence rates for the two groups. If there is no overall difference between the incidence rates of the placebo group and the incidence rates of the vaccinated group, then the efficacy would be 0. If all the subjects in the vaccinated group would be fully protected, then the efficacy is 100%. On the left-hand side, you see a grid of plots with biomarkers values on the horizontal x-axis and the reported incidence rates on the vertical y-axis. The various plots show the data for different populations A, B, C, and different disease severity level 1 to 3. The size of the points indicates the size of the trial. A single network MBMA model was built to describe all different vaccines and account for all populations and disease severity levels. In that way, all data points contribute and strengthen the biomarker incidence rate relationship for each population and disease severity level. On the right, the MBMA model shows the relationship for population B at disease severity level 2, which all literature reported data points scale to these situations. The model shows that as biomarker value increases, the incidence rate decreases. The yellow shadow is the confidence interval of the model. Then this population B was simulated obtaining a range of biomarker values when treated with vaccine X or with placebo. Based on the calculated incidence rates, the efficacy for a study with vaccine X was predicted to support a go/no-go decision for this program. So the MBMA model has provided a very powerful overview of all vaccines against virus X that have been studied so far and allowed to position for the new vaccine among them. This approach was also applied to a RSV antibody that was designed for prophylactic use, which is referenced here. The fourth example shows an analysis where immunogenicity data were extrapolated to assess long-term protection by vaccine against Japanese encephalitis. The first model was built to describe the antibody change over time. The model was subsequently used to predict the duration and extent of the serological protections. A piecewise linear mixed model was chosen because it provided the best fit to the model. The antibody declined from the observed peak. Individual seroprotection was based on the accepted threshold that the antibody titer should remain above 10. Immunogenicity data from randomized clinical trial using 1 or 2 doses of the vaccine in healthy volunteer was measured at various time points up to 5 years. The box plot shows the observed antibody titers between time is 0 and 5 years in subjects receiving a single dose of the vaccine. The green lines represent the accepted threshold of protections of 10 times dilutions. The plots on the right-hand side summarize the protection against the virus we can expect based on the titer observation and threshold. The spaghetti plot shows the population predictions in black line and individual fits in blue lines of the antibody titer of 99 subjects using the piecewise linear model. The population average closely matched the observed median antibody titers observed up to year 5. The red segment indicates the observation time period and the blue segment indicated the extrapolation period. The plots on the right-hand side shows the predictions of the proportion of subjects that would be seroprotected through time with 95% confidence interval based on model parameters uncertainty and individual variability. The open circle represents the observed seroprotection values, using the assumption that the log linear rate of antibody continues after 5 years. The majority of the subjects were predicted to be protected for at least 10 years after single-dose vaccination. This example illustrates simple model can also allow prediction for long-term immunogenicity prediction for at least 10 years. As data emerges, the model can validate the extrapolated period to validate the assumptions of the model that a linear model is used. The work was also extended to extrapolate to other populations as well. The final example is to assess whether QSP can further support the understanding of immunogenicity. In the top left diagram, with all the existing virus and most recent with COVID-19, we have more understanding of the factors that contributing to the increasing, shown in red, and decreasing, shown in green, of immunogenicity. For example, binding to DC cells will increase immunogenicity while depleting of B cells or certain co-medication will decrease immunogenicity. With all this knowledge, the B-cell memory model shown in the previous slide can expand into a QSP model, which you can see in the schematic on the top right, by incorporating in vitro, population, and clinical data. The developed model will allow us to gain insight through population simulation of the potential optimal dose, regimen, and route. In addition to gaining more understanding of the mechanistic insight of the mode of action of the vaccine and the identification of useful efficacy and safety biomarker, and thus differentiate factors that influences the differences in response in various populations. The bottom left plot shows an example of the identifications of optimal interval between the first and second dose using vaccine efficacy response. So in conclusion, we highlight that vaccine will need to be safe, but also we should not forget we also need efficacies in vaccines too. The urgency of the medical need is too high to not answer the key questions, such as do we have the right dose and regimen to get optimal level and duration of prophylactic protections, and if not, do we need to revaccinate it or use initial booster regimens, and do all the subpopulations such as elderly and pediatric and people with underlying disease, aka with comorbidity, requires the same regimens. We also hope to demonstrate that vaccines too will benefit tremendously from model-informed drug discovery and development techniques and via model interconnectedness as well to answer these questions, especially using model-based meta-analysis, which is a powerful tool to translate immunological response to prophylactic protections and conduct comparative effectiveness assessment in our search for the best vaccines for global scaled up and rolled out. Quantitative system pharmacology approaches established for unwarranted immunogenicity of biologics can be repurposed to predict warranted immunogenicity response of novel vaccines and describe the mechanism of novel vaccine platforms. All of these examples and ideas are great foundation for future work, and this is very important for us to prepare for any future pandemic situations or emerge of new viruses, or bacterial or parasitic. By bringing these state-of-art solutions to the table now, we can jointly ensure we can tackle the future pandemic or different disease which will require vaccines using quantitative approach with MIDD and model interconnectedness. And finally, on behalf of Certara and Critical Path Institute, we thank you for watching this module, and we would like to acknowledge Craig, Jeff, and Mark. And this concludes the presentation. And goodbye. Have a great day.